everybody. I'm Bobby, back again with you on the daily lessons and preparations for the Csir-Net Lifesciences and Neet biology. I hope you listen to my lectures, take down notes, and prepare for the best. Wish you all the best. Best wishes. Keep track of my lectures. I'll be starting right now, okay? Bye.
Right, continuing with the lessons of yesterday on photosynthesis, we have Crassulacean acid metabolism, that's CAM, Crassulacean acid metabolism, right? This pathway is found in succulents, mostly members of the Crassulaceae, as bryophyllum and sedum. Right, and a few members of Bromelaceae, such as pineapple. It is a device designed to meet the pressures of heavy transpiration arising from the xerophytic environment. These plants obtain their CO2 requirements during the nighttime when they keep their stomata open, and as CO2 buildup occurs, the cell sap turns acidic. This process is called as dark acidification. Remember these topics, they are effective for your MCQs. Right? In the following daytime, the stomata remain closed, minimizing transpirational losses. But with the advent of light, the carbon dioxide absorbed during the preceding night is utilized for photosynthetic purposes. The process of light deacidification then occurs. Thus, there is a time lag between absorption and reduction of carbon dioxide. This arrangement helps to lessen transpirational stress, but is responsible for the extremely slow growth of these plants. Next will be the factors influencing photosynthesis. Carbon dioxide concentration of the atmosphere is 0.03% or 300 parts per million. It is a limiting factor, right, for the available carbon dioxide. And the concentration is lower than the optimum for high photosynthesis. And increase in its concentration up to 0.1% at 1,000 parts per million increases the rate of photosynthesis. In most lamp plants, a decline is observed beyond it. Light intensity. The light intensity at which a plant can achieve maximum amount of photosynthesis is called, saturation point. Its value is 800 to 1,000 feet candles. That's about 10% of full sunlight in shade plants, 50 to 70% of full sunlight in C3 sun plants, and up to 200% of full sunlight in C4 sun plants, example sugarcane. Right? The next subject will be the quality of light. The maximum photosynthesis occurs in blue light and red regions of the light spectrum, but most of the absorption is carried out by chlorophylls. Minimum photosynthesis occurs in the green wavelengths. And the fourth would be temperature. It does not influence light reactions of photosynthesis, but affects an enzyme called controlled dark reactions. The minimum temperature at which most plants start photosynthesis is 0 degree to 5 degrees centigrade, but it can be as low as minus 20 degrees centigrade for lichens and 35 degrees centigrade for some gymnosperms. The maximum temperature at which photosynthesis can occur is 55% in some desert plants and 75 degrees centigrade for hot spring algae. The optimum temperature is 10 degrees to 25 degrees for C3 plants and 30 degrees to 45 degrees centigrade for C4 plants. And next will be oxygen. The small quantity of oxygen is essential for photosynthesis, except in some anaerobic bacteria. C3 plants show optimum photosynthesis at low concentration. The possible reasons are: 2. Oxygen takes part in oxidation of photosynthetic pigments, intermediates of enzymes, and enzymes in the presence of strong light photooxidation. Oxygen is a strong quencher of excited state of chlorophyll. Oxygen competes with carbon dioxide producing power. However, this effect is not known in C4 plants. It converts RuBP carboxylates to RuBP oxygenase at very high oxygen content. The rate of photosynthesis begins to decline in all plants. The phenomenon is called Warburg effect. Water supplies hydrogen and electrons for carbon dioxide fixation. However, less than 1% of the total water absorbed is utilized in photosynthesis. The rest is lost in transpiration. Even a slight increase in transpiration reduces the leaf hydration that cuts down photosynthesis by causing stomata closure and hence decreased CO2 absorption, loss of leaf turgidity, reduced absorption of solar radiations, and decreased enzymatic activity. Thus, photosynthesis is more sensitive to dehydration than any other metabolic process. Air pollutants. Dust and smoke particles present in the atmosphere reduce photosynthesis by reducing light penetration and forming a layer over the plant. Sulfur dioxide, nitrogen oxides, hydrogen fluorides, and other air pollutants also decrease photosynthesis. 8. Minerals. Magnesium is a component of chlorophyll. Iron and copper and manganese are required for synthesis of chlorophyll. 2. Manganese and chlorine are linked to photosynthesis of water at PS2, photosystem 2. P as phosphate is essential for ADP synthesis. Enzyme activators of photosynthesis include potassium and sulfur. Low availability of any of these minerals reduces rate of photosynthesis, right? Now, factors influencing photosynthesis: carbon dioxide. Carbon dioxide concentration of atmosphere is 0.3 or 33 ppm, as already told you. Now we're going to the next part, which is the next topic. We've just finished on photosynthesis. We're going to F1, enzymes, basic concepts and principles of catalysis. Enzymes, the catalysts of biological systems, are remarkable molecular devices that determine the patterns of chemical transformations. They also mediate the transformation of one form of energy into another. The most striking characteristics of enzymes are their catalytic power and specificity. Catalysis takes place at a particular site on the enzyme called the active site. Nearly all known enzymes are proteins. However, proteins do not have an absolute monopoly on catalysis. The discovery of catalytically active RNA molecules, ribozymes, provides compelling evidence that RNA was an early biocatalyst. Proteins as a class of macromolecules are highly effective catalysts for an enormous diversity of chemical reactions because of the capacity to specifically bind a very wide range of molecules. By utilizing the full repertoire of intermolecular forces, enzymes bring substrates together in an optimal orientation. They prelude to making and breaking chemical bonds. They catalyze reactions by stabilizing transition states. The high energy species in reaction pathways. By selectively stabilizing a transition state, an enzyme determines which of several potential chemical reactions actually takes place. Right? Characteristics of enzymes. Enzymes accelerate reactions by factors of as much as millions or more. Indeed, most reactions in biological systems do not take place at perceptible rates in the absence of enzymes. They do not change the equilibrium. The specificity of an enzyme is due to its precise interaction with the substrate, with the enzyme. This precision is a result of the intricate three-dimensional structure of the enzyme protein. Like catalysts, the enzymes do not start the chemical reaction, not change its equilibrium, but enhance the rate of reaction. Enzyme lowers the activation energy or energy required to overcome the barrier of the reduction. Enzymes are not imported. Every cell produces its own enzymes. They may be active inside the cell's endoenzymes or intracellular enzymes, 70% in mitochondria, or outside the cell exoenzymes, etc., digestive enzymes. Amylase and protease enzymes are now added like detergents for washing, and Rene tablets are used for preparation of cheese. Enzymes have a high molecular weight, 6,000 kD for bacterial ferredoxin, 250,000 for catalase, 480,000 for urease, and 4,600,000 for pyruvate dehydrogenase. Therefore, they occur in a colloidal state. Optimal pH for enzyme action varies for enzyme to enzyme. Example 2 for pepsin, 6 to 8 for salivary amylase, and 8.5 for trypsin. Most intracellular enzymes work at a neutral pH. Enzymes are thermal labile. High temperature denaturates enzyme. Optimum temperature of enzyme activity is 30 to 40 degrees centigrade for most of animals and 20 to 30 degrees centigrade for plants, within limits. Q10 is equal to 2 to 3. The first ribosome discovered by Czech et al. In 81 in tetrahymena was self-splicing group 1 intron. It removes introns, formed newly synthesized RNA. The second discovery was RNase P, ribonuclease P, Altman et al. 92, which separates tRNA from snRNA at the 5' end. Nolan et al. 92 discovered the prokaryotes' peptidyl transferase is not a protein but a component of 23S RNA. There are all questions that come for the MCQ. Lysozyme is an enzyme that hydrolyzes, breaks down the cell walls of bacteria. Lysozyme is very common. It is found in saliva, in digestive fluids, even in body secretions. It is one of the first lines of defense against bacterial infection. Lysozyme is a small enzyme, a single polypeptide chain with both the alpha helix and beta sheet regions. And now for the major classes of enzymes, we have 6, that's section F2, 6 major classes of enzymes. Class and the type of reaction. The first would be oxidoreductases, which gives the type of reaction called oxidation reduction. Transferase, which causes a group transfer. Hydrolysis, that causes hydrolysis reactions, transfer functional groups to water. The examples of the first one, lactate dehydrogenase, second was nucleoside monophosphate kinase, NMP kinase, and the third hydrolysis would be chymotrypsin, and lyasis is additional removal of groups to form double bonds. Example with fumarase. Then you have isomerase, which causes isomeration or intramolecular group transfer, and that's caused by triose phosphate isomerase. Last is the ligases, where ligation 2 substrates at the expense of ATP hydrolysis. That's caused by aminosyl tRNA synthetase. So tomorrow's topic would be F3. Many enzymes require cofactors for activity. I'll be going on to the MCQ session or the sections now. Alright, yesterday we stopped at number 53. Today is number 54. A 30 residue peptide was treated with trypsin and the peptides were separated by HPLC. 4 peaks A, B, C, and D were obtained. Peptides corresponding to A, B, C, and D were reduced and alkylated selectively at cysteine residues. The sequence obtained from A, B, C, and D after reduction and alkylation were A, AEK, B, C, that's S-alkyl, EPG were R, and WC, S-alkyl, SPPK, and C, S alkyl, C a GGK, D would be SC, Salcyl EAFC, and the sequence of the 30 residues peptide is, just magnifying it, check which is the right one, take screenshots of it and guess the answers. Okay, the next question would be, an alpha helix in a peptide or protein is characterized by hydrogen bonds and characteristic dihedral angles. Choose the right combination. Hydrogen bonding between the amide, CO of residue 1 and amide NH of residue 1 plus 4 dihedral angles in the regions theta equal to minus 50 degrees and psi equal to minus 60 degrees centigrade. Hydrogen bonding between amide NH of residue 1 and amide of CO of residue 1 plus 4 dihedral angles single in the region of theta equal to minus 50 degrees and psi equal to minus 60. And hydrogen bonding between the amide CO of residue 1 and amide NH of residue 1 plus 4 dihedral angle in the region of theta equal to minus 50 and psi plus 60. And hydrogen bonding between the amide CO of residue 1 and amide NH of residue 1 plus 3 dihedral angle in the region of theta equal to minus 50 and psi equal to minus 60. Last question will be, precursors of the atoms in the purine skeleton are. Here are the diagram. Have a look at it. Okay. Can you see it? Yeah. We're going back to the answers. N1 ASP, C2 and C8 formate, N3 and N9 guanidine, C4 C5 and N7 glycine, C6 CO2. Next answer would be N1 ASP, C2 and C8, citrate N3 and N9, amide nitrogen glycine, C4 C5 and N7 glycine, C6 CO2. N1 ASP, C2 and C8 formate, N3 and N9 amide nitrogen of glycerine, C4 C5 and 7 glycine, C6 CO2. And the last is N1 glutamine, C2 and C8 acetate, N3 and N9 amide nitrogen of ASN, C4 C5 and 7 glycine, C6 CO2. Well, I'll read this too and then we'll wind up, I guess. It has been observed that in 55 to 10% of the eukaryotic mRNAs with multiple AUGs, the first AUG is not the initiation site of such cases. The ribose skips over one or more AUGs before encountering the favorable one initiating translation. This is postulated to be due to the presence of the following consensus sequences. I hope you can see the sequences. Which of the following sequence sets related to the above postulations is right? A and B, A, C and D, B and D. Right? Next question will be the value of Tm, midpoint of denaturation, delta HM enthalpy change at Tm, and delta Gp, constant pressure, heat capacity change is delta Gp. Others free energy delta G. A protein are measured in differential Canning calorimeter. So delta Gp at temperature is the Gibbs free energy change at any temperature Tk can be estimated using the following form of the Gibbs-Helmholtz equation, but the values obtained from these measurements. The stability curve for the protein simulated using the above thermodynamic values is given below. See the diagram carefully. Right? Temperature, delta G there, the free energy. The shape of the stability curve is due to 1. A. Hydrogen bonding, electrostatic interactions only, van der Waals and electrostatic interactions only electrostatic interactions, and only hydrophobic interactions. So I'll see you next class. Bye.